Hi, how are you? Morning.
Okay. Good morning. Yeah. Slept in a bit.
Oh, yeah. How are things going for you?
Pretty good.
Good.
Yeah. I got one more physical thing to do. I have to see if I can ride a bike again.
Oh, good.
Put my pair of shoes on first.
Yeah. Hello, Ujjwal. You there?
Hello, hi.
Hi, how are you?
For the last 3-4 days, I'm not feeling very well. I have very mild fever
Oh,
this last 3-4 days. Now coughing has gone, but there is still 99 to 100 Fahrenheit fever.
oh. That doesn't sound good.
Yeah.
Hopefully, hopefully you can get some rest. A little bit.
Yes, I'm taking rest. Yes, I'm taking complete rest. Today, I'm much better than yesterday. Yesterday, there is a lot of coughing. Today, coughing has gone completely.
Yeah. Well, that's good. Well, Susan and Jesse and Abhishek and Mayukh.
Okay, I think you can hear me.
Yeah.
Can you hear me? Yeah, if you can see me, you can't hear me, but if you can, I don't know. The camera and the sound seem to work off the same circuit in this computer.
Yeah.
I may need a new computer.
Oh.
Sometime.
Maybe. I don't know, but yeah. Well, I mean, we can hear you. That's good. Welcome. So welcome to the meeting, everyone. Again, hi, Jesse. We'll have some updates from Ujjwal and Mayukh in a minute. I just wanted to go over maybe some other things that are going on. We just submitted a paper for Bacillaria behavior, so we submitted the abstract for that, and I'll talk about that a little bit later. It's for another book chapter that's being written, the proposal, or kind of more of a outline, I guess. I don't, it's just based on what we talked about previously, and so I'll go over that in a bit. Also, if anyone has any news on their side, now's the time. Anything going on new? Might be of interest to the group.
I don't know how. I think you probably already know this, but just to say this for the group. I'm definitely looking at the 2 papers that I mentioned in the DevoWorm Slack channel. I'm interested in kind of looking at those papers again. I'll be starting a summer school later today too, which will be interesting on brains, minds, and machines. I don't know if I'll feedback here much, but I'll probably talk about it next week after it. And lastly, there are a couple papers in around that kind of the match point of neuroscience and cognitive science that Bradley mentioned one that I probably will go over in the future, and there's also definitely I think I made an issue for it in the Slack projects board about the biology at scaleless, something like that paper.
Oh, yeah.
I definitely in that paper in particular. So I might go over that in the future, but not this week.
Yeah. Well, that's good.
That's all.
Yeah. Susan, of course, Susan and we were talking about the axolotl embryos last week. So any other additional thoughts on that, or?
About the axolotl embryos,
Yeah.
I haven't done much this last week because I was kind of given a reprieve on my thesis writing for the week, so it was sort of an off week.
Yeah.
Trying to straighten out my office partly. I would like it functioning properly for September, and that includes having my 3D printer working so that I can make a ball microscope.
Oh, wow.
Anyway, so yeah, it was sort of an off week. I haven't done much. As to the chaos papers, I don't know whether I'm going to use it in my thesis or not, but it is a method of measuring things that seem unmeasurable. And so that's why I thought you'd be interested, because it is a measure of behavior and of how fractal something is, like how fine a detail something, well, how many branches something has. It's, I don't know, diffusion limited aggregate type stuff. So it is relevant to looking at something and seeing whether it has a deterministic behavior.
Yeah. We haven't talked too much about chaos, and especially diffusion limited aggregation. It's probably something we should talk about, though. And especially with the Bacillaria paper, we'll talk about that in a bit. Maybe that's useful there too. But yeah, thanks for those papers. We might go over in a little bit more detail later on in the meeting. And then let's see what our chat says here. So Mayukh says, hello everyone, my laptop charger isn't working, so I'll be attending this meeting on my phone. I won't be able to share my screen today. I think I got some of the stuff you put up on Slack though, Mayukh. I backed it up just in case, so I can present that. I can share the screen and then you can see it and maybe talk about it a little bit. Is everyone still there? Okay. All right. Yeah, I just want to make sure I don't. I think Dick dropped off, but I wasn't able to. I don't know if everyone still can hear me or not. I just wanted to make sure.
I can hear you. Yeah.
Okay, good. Okay, so why doesn't Ujjwal start?
Yeah, I can hear you.
Okay, thanks. Yeah, why don't you give an update?
Yes, friends. So since last week, I doesn't have done a lot of work on the project due to my health issues.
Yeah.
So what I have started doing is documenting my code, whatever that I have, on the CSV and all these things. I start writing a document, a Google Doc, which I will be sharing in 3-4 days. That has all the things, like it's a kind of weekly for the contributors who are coming, so that they don't have to go and search here and there. They just have to read that. It has all the information, like from which tag or which thing is being changed in the portal. So I have tried to give that amount of detail in that. Other than that, I have created a form, I guess, for deciding the name. So I haven't got much reviews from on the DevoWorm and from the people here, but I share the same form in my research group as well. After deciding a lot, I came up with a name C Elegant DevoWorm .heroquap.com. Because you can keep a uniformity, yeah, this is the DevoWorm model, but since they have different kind of models, so these can go in the first name. So I will be hosting it in a day or two. Other than that, that's the update from me.
Well, it sounds good. Yeah, we talked about that in last week's meeting about the name of the different apps. So I guess that sounds good to me. I mean, like if we had like the name of the organism and then DevoLearn after it, like whatever it is dash DevoLearn, something like that. And it seems like it's pretty consistent with the naming convention.
Yes.
Yeah. So Mayukh said he's on his phone today, so let's see if I can get pull up what he was sharing. So actually, let me present my screen.
I could just tell what it is. Like,
Yeah.
yeah. Yeah, it's not much anyways. What I've been doing is that I've been like testing out my own library to look for errors and like faults. And I didn't find any until now. Other than that, I've just been exploring the data, and I've been like looking for patterns like in the growth process. Like I don't know much about the cell biology part, but I've been trying to find like in my own mathematical approaches. Like I think Bradley will show that GIF which I sent to him on Slack. I think you could show that one. It's like I'm trying to map the distances between the centroids of each cell within the embryo, like with time. I'm trying to plot them in a matrix. Like I'm basically what I'm doing is that I'm extracting the centroids using my own library, and then after extracting the centroids, I'm mapping out the distance between, like the distance matrix, basically, like the distance between each node, like in a square matrix. Then like I'm just trying to find pattern, just exploring. Yeah, so I think Gary could show that GIF. It's on Slack, I guess.
Yeah.
There were, I guess, 3 GIFs.
Yeah. Okay, let me see if I can present now.
Like using the library, we can make a map of like nodes and like lines which represent the cells and the like centroids and the connections between them.
Yeah. Yeah.
Kind of interesting. And even Bradley had a paper which he sent me, so I've been trying to read and understand what's going on in there. It's kind of going over my head, but I'm trying.
Well, we'll talk about it a little bit. So let's see. First thing I wanted to talk about was this. So the DevoLearn umbrella. So it came up between Ujjwal and Mayukh and myself, asking the question like, how do we name? What is the convention for everything? So this is what I'm kind of envisioning here for the umbrella. So the main GitHub organization is called DevoLearn. And then the software here, Mayukh's pre-trained model would be DevoLearn 0.9.1. And of course, that version number would change with a new version or whatever. But that would be that part. And then over here, we'd have the species-specific model. So this is the thing that Ujjwal was talking about with like the species name or the organism name dash DevoLearn dash or DevoLearn Heroku app, whatever. And it would be just separate Heroku apps for each of them. So you'd go, DevoLearn would contain the links to those apps, which would be hosted on Heroku app because you can't host something like that on GitHub. And so that would be the sort of the implementation there. And, they all have to have their own names since they're separate entities. And then finally, the DevoZoo, which is, we haven't talked too much about, but it would be sort of over on this side and also accessible from the DevoLearn umbrella. So when I say umbrella, some of you may not have been here last week. Let me see. This is the tab. So last week, over the last week and a half maybe, we started a new GitHub organization, and that is called DevoLearn. So this is just, in GitHub, you can start an organization if you want to have like a face for your project or a face for your organization that's distinct from what you're doing. I have one for DevoWorm, but that's actually, I wanted to keep this a bit separate, just so that people can find it and join the organization and things like that. So this is a new organization. It has a couple of repos already. These are the repos where Ujjwal and Mayukh have been contributing. We have the DevoLearn, which is the pre-trained model, the C. elegans DevoLearn, which includes a lot of what Ujjwal's been working on, the contribution guidelines, which are just basically how to contribute, the media, which is a full, images and other things like that. And then this new one I made called Data Science Demos, which is going to be for people to create demos in this data science analysis space. So Mayukh has been working on this network stuff, which we'll talk about in a minute, and he can push there when he's ready. But then also Krishna Katyal asked me if he could contribute something as well, and it's I can't remember exactly what it is. It's very data sciencey, so I said sure. Just commit it to this folder. So this is going to be the folder if you have any demos you want to put up that are germane to analyzing data or something of that nature. If it's like a tutorial in like a notebook or just like some methods or like some visualizations, this would be the place to push them. And we'll organize this, later on by topic. The networks one was pretty obvious, but so when Mayukh gets a chance to push to the networks folder, he's going to create, one of the things he's going to create is he has this network simulation, which we'll see in a minute, and you can download the centroid positions and the distances between the centroid positions, and all of that would be in a CSV file. And then what you can do in GitHub is you can upload a CSV file. If you just have the raw CSV format, you can upload that file to GitHub, and it'll actually, and I don't have it in this table, it'll render a table with the numbers in it. So it's as simple as, if you have something called CSV, you can say something like AB, and then 1, 2, and then we can commit. And then it creates this table that you can actually you can't interact with it, but you can download everything in CSV format, and it creates a nice table for people to browse online, and then we can use that as part of the.
I won't just upload the CSV file. I'll also upload a link to the executable collab notebook so that people can play around and it's all made on DevoWorm, so they can just get a taste of the library and they can also play around with their own video files. What I'm doing is with my own video file on this Embryo. Maybe they have some other video. Maybe they want to use that, so they can do that.
Okay. Yeah, we can put the
it will be an executable runtime, not just something that they can download and just. It will be something that people can use and play around with.
Right. Yeah. Right. So we have the so we would put the collab notebook here as well. Push that, and then you can open up the collab notebook, and I think it works best if you actually open it offline, because in GitHub it doesn't open directly all the time. So if people want to use it, it'll tell people how to open it up. But I like the way collab notebooks work. You can open them up, and then it's connects directly to your Google Drive, and it's nice. So we have a comment here. Jesse says, when you see an organization on GitHub, it's something specific to join or contribute like normal GitHub. Oh, so this is it's like normal GitHub, except that on our end it's an organization instead of an individual. So I think you can join the organization. You can't follow the organization. There's some rules like that, but it's basically like OpenWorm is an organization. If people are familiar with the OpenWorm GitHub, you can't do certain things, but you can definitely contribute to it. Anyone can contribute to it through a pull request, but you can also join as an editor if you're contributing a lot. Like Mayukh and Ujjwal are contributors right now, and if you want to become a contributor, then I can add you on. But this is if you're doing a lot of uploading of stuff and manipulating stuff. So I would encourage you to check this out. the DevoWorm organization. See what everyone thinks. There's a lot of stuff. We're going to keep adding things on over time. So this is meant to be very flexible, and we can actually. Another reason why I wanted to create an organization for it too was because it's it's sort of a standalone thing, so you don't have to go into the DevoWorm GitHub and look around and try to find things. So actually, this was I think Ujjwal's suggestion or Mayukh's suggestion to create an organization. So that's the DevoWorm. We talked about the umbrella. We talked about the logos last week. Now I think I'll go on to talk about the Bacillaria papers. So this is kind of a shift. We'll talk about the GSoC stuff more next week. But I think Mayukh and Ujjwal have done a great job on the DevoWorm organization. And I think we'll probably launch that like at the end of GSoC formally. So I think we mentioned it a little bit to people, but it's not, formally launched. So we'll try to launch it probably by the end of GSoC. And that means like we'll just announce it formally, might do a blog post, and just kind of formally announce it to people so that the OpenWorm annual meeting's coming up in September, so I'll probably get a good mention there. So congratulations on that. Any other questions before we move on? Oh, actually, I did want to talk about Mayukh's network stuff before the Bacillaria stuff. So I did download Mayukh's stuff with the Embryo Networks. So Embryo Networks are something we've been doing in the group. Like we did this stuff a couple years ago from the same data set that Mayukh is working with. But he's doing he's actually doing it a little bit differently. But maybe not. Let's see. So he's generated these animated GIFs. So this is a basic network. These values along these two axes, I imagine. So the data set that he's working with, the Epic data set.
Yeah, they're just there is the dimensions of the image. Like this is just an overlay over the image. Like these are the location of the nodes, like the centers of the cells.
Right. So the Epic
Scale
data
is just
set.
like the size of the image.
Yeah. The Epic data set actually gives position values in like sort of the space that's undefined. So it just gives you these coordinates. One thing
the Epic data set actually.
What was that?
This isn't the Epic data set. This is another a small data set which I found.
Oh, okay.
Online.
Right. So this
I could use I'll send that link.
All right.
Later on, maybe not now.
Okay. Yeah. So okay. So then maybe this doesn't apply. Maybe it does. But when we were using the Epic data set, they give you these coordinates, but they're not aligned across embryos, just kind of like numbers. So I did a normalization procedure for all of the embryos where you center the coordinate system on the middle of the embryo. And then it's kind of like where the P0 cell is, the centroid of the P0 cell. And then you can do like a z-score and some other transform to get that number. And then you have a unified coordinate system. And it's still something that I think I was referring to as embryo units. Although it could be, you could, I think, map that to an actual metric measurement. So, but the idea is you have these centroids of cells, and they're in a certain location in XY or XYZ space. And then as the cells divide over time, you get new cells, and they come up in the same coordinate system. Then each of these cells are connected somehow by a distance. And so you have these cells that are connected by distance in this network forms if you have this fully connected network. And then you can evaluate the strength of these edges by, some threshold. You could use, actually, you can use a bunch of different criterion for these connections. Like these edges could be weighted by their distance. They could be weighted by maybe some hypothetical cell signaling.
Yeah.
And so,
yeah, for now, the distance matrix that I'm making, I'm currently only like, these lines are just like drawn, okay? So I think it would be better if you show the next GIF. Like, there was a GIF, I guess.
yeah.
That would
yeah,
give a better idea because this one's pretty dull.
Yeah.
This one was made during the preliminary stages.
So, I mean, this is based on just the distance between the cells, though. I mean, this line is, you could measure it out. Yeah.
It's just a mapping of the cells.
Yeah.
I think the GIFs would give a better idea.
So this is the GIF. So this is basically what happens in the embryo where, I mean, this isn't a real embryo. It's just like cells kind of appearing.
Exactly. From a real embryo, I use the DevoWorm tree to extract the centroids from a real embryo and then I map them like this on a black photo.
Oh, okay.
Like, just to make it look clean.
Okay.
So
So
this
this
is
is
from an actual embryo.
based on
I think there's another GIF which will visualize it even better.
Okay.
I think there was another GIF. No, I think the one on the bottom, I guess. If I remember,
Oh.
yeah, this one. Like this is how it was extracting the centroids.
Okay, yeah, this is extracting the
this-
centroids. Yeah.
These are the same circles which are there like on the other GIFs.
Okay.
It's just that I removed the background and I replaced it with a black background to make it more clean. Yeah, this one gives it off. Like, yeah.
So this
So-
is the,
What I was like, by seeing this GIF, so network mapping. So basically, what I'm getting is like he has right now connected all the centroids with all the other centroids, like in a form of a bipartite graph. So rather than just like connecting it with by using brute force, like I was searching through the data, and the sir has provided me a long time ago OSI data, I guess, and there is a very good video of embryo in which the cells are labeled as A, B, C, D, A1, B1, C1. I don't know like what's the biology behind it, but they are labeled somehow.
Yeah,
And there is double. Yeah, so they are double. They are double A's and all these. So like if we can map using these in some sort of.
Look, I'll tell you. What DevoWorm does is that it can extract the cells for you. Like it can tell you where the cells are. But I cannot, like the DevoWorm cannot determine the lineage. Like they are basically, if I'm not wrong, correct me if I'm wrong. Like in the initial stages, there are there are two basic lineages. That is the AB and the P.
Yeah.
DevoWorm cannot identify which is AB and which is P, but it can identify where is like the cell.
Like I'm not like talking about like something to incorporate in DevoWorm itself. Like it's like a paper that we are focusing on. Like if we can do something like that, it doesn't have to be like totally integrated with
We have to
DevoWorm.
manually annotate it like this is AB and this is P.
Yeah.
Manually.
Yeah.
It is
For the
possible,
paper purpose, I'm talking about, like not for library.
yeah.
It would be possible.
So this
I
is
was just
the,
trying to map out on the distance matrix, like it looked interesting.
yeah. So this is the distance matrix here in Smooth. So you have all these sort of components for a network. So I was
The
gonna,
distance matrix basically shows the distances between each node from the other.
yeah,
So if there are like n nodes, like you notice how the square, like the matrix gets larger and larger with time
Yeah.
because there are more nodes. Like
Right.
if there are n nodes, there are n cross n. Like that is the size of the matrix. So that is how it goes.
yeah.
And I just tried to smooth it to look for some like broader patterns, but it wasn't much of a success.
Yeah. Well, I
Interesting
actually,
anyhow.
I took some slides out of some presentations that I've done on this, so I can send you those presentations if that's something you want to explore further. But so this work with Embryo Networks, this is something that Dick and I and some of the other people from the been around the group longer have worked on. I presented on this a couple times actually, this kind of work. So the idea behind embryo networks is that we have these cells that divide and they form this embryo, but you can characterize it using a series of sort of edges. You can represent it as a graph. So you can have the centroids of cells be these nodes, and then their distance be edges, and create these graphs. And what you want to have is this sort of 5-dimensional representation where you have 3 dimensions of space, 1 dimension of time, and then this other symbol, which is some context Q. In this case, it's I, so that's the distance along the AP axis of the lineage tree. But you can use other things for I. You can use like some sort of, if you're interested in signaling, like an interactome between the cells, you can use some information and use I as the parameter to represent that. Or there are other things you might be interested in. This is a network that was made of the C. elegans embryo from the extracted epic data set. And so this is very much what they call a hairball in network science. So it's just like nodes that are proliferative, and they make these nice pictures, but there's got to be some way to, sort of analyze what that means. But in network science, people use these hairballs to sort of show the complexity of something. And so this is like all of the time points overlaid upon one another. So this is kind of like where you have sort of a lineage tree emerging, but also the distances between cells, and it forms this hairball. This is sort of a subset of nodes shown in a 3-dimensional space. So you can get a sense of like what it looks like when it's not a hairball, when you have this local neighborhood. And then, you can use this. This is the paper I sent to Mayukh, and this is, of course, a paper that we have up on the website.
I had a question on this. Actually, could you scroll up?
Yeah.
Yeah, just a bit. Yeah, just there. Yeah, I can see a 3D graph to the left. So how did you guys annotate that graph? Like, how did you guys know that this cell and this centroid is like it belongs to this lineage? Like, how did you guys determine that?
Well, it's
is there some kind of is there some specific approach? Like, it can be automated in any way.
No, the epic data set comes in 2 flavors. So one is the images, which allows you to extract cells, but they're not annotated. And then the other one is a set of files that has like the distances and the names. So like that's not an image file. That's like a more of a CSV file type thing. And so that they have those data that you can get, but you can also, that's not image data. So, it's just based on their like cell tracking that they have. And so that's where we got that from. Each cell has an identity in C. elegans anyways. And so you have the name of the cell, and then you have the position, and then you have to average over the embryos that they give you, and it gives you this position. You can estimate a position for, any stage. So that's they have those data, and that's separate from what you're working on. But of course, using, say, something like machine learning versus just the sort of cell tracking that they're using is going to give you.
I remember now. Actually, I worked on the same data from a dataset, but I didn't map it out. Like, I made this deep learning model long back. It was during phase 1 of GSoC. Like, it could take the position and time and size of the cell, and it could determine like the lineage. It was at a very like 99 or something accuracy. But I just kind of dumped it out later on because it wasn't very relevant at that moment.
Yeah.
But right now, what I can think of is that like using DevoWorm as a backbone, it's actually possible to like map out a 3D node map or the 3D centroid. Like, of course, like we have a 3D image of an embryo, right?
Yeah.
We can pass that 3D image through the model in DevoWorm, and we can get a list of centroids like X, Y, and Z, which can be used to map out nodes just like this. And then we could use that kind of node map to like train a model. Let's see, like I will explore on this. Even though it's not related to GSoC, like
right.
Let's see. Like what DevoWorm that it's automating the part of making the node map from videos.
Right.
That's what it's doing. But it doesn't know what node is from which lineage. That's what we can use epic dataset to determine that.
Right.
But for that, we need normalization, I guess,
Yeah.
because different from like embryo to embryo. Yeah. So
Yeah. So this is the interactome, and then that's just kind of more of the same thing. So then there's this question though that exists, which is when you get these networks, they look like hairballs, but they actually have structure in them. So you were showing those matrices, and we can take these networks and create matrices like you did, but we can also analyze those matrices in ways that tell us what the structure of the network is. And so you can see here this is from a paper on the sensory motor connectome and zebrafish, that you have different patterns across this matrix. So, you have something called a small world network where you have, they define it in the literature. I don't want to get too much into what these exactly are, but you can see in the matrix there's a certain pattern where most of they basically threshold the connections by some criterion, and then they find that there's this diagonal sort of behavior. A scale-free network is sort of where there's clustering, but not diagonally. It's maybe along the edges here. And then the random condition where there's just significant or edges above threshold across the matrix. So this is something you can do as well. You can do these sorts of tests to ask what kind of a network is it. And so this was done with the C. elegans data versus a random embryo, and it's definitely significantly different from a random embryo. We don't really know. We know in the C. elegans connectome that it's sort of has a small world aspect, but in terms of the entire embryo, we don't know. And again, these embryo networks are somewhat different than the connectome in the brain because we're using a different criterion. We're saying it's just distance based, and but there's definitely information in here. And so you can look at it like this. You can look at it in terms of like sort of the anatomy. So in a C. elegans, this is C. elegans right before it hatches out of the egg. You can characterize the cells kind of in this spiral network. You can also take a mouse blastocyst, and this was based on a mouse data set that, if you take the cells and you use the same criterion, you can map, essentially approximate this phenotype here. And so, again, it just shows you like aspects of the phenotype, but there is probably information in there. We talked about topological data analysis a couple meetings back, and that's kind of what we're doing here. We're not really doing it in the same way that they do it in the literature, but it's a similar idea. So you might have like motifs within these networks that are just like kind of an area within a bunch of nodes and their connections, and you can measure those those little geometric areas, and they might have some significance to spatial organization, things like that. So we don't really know what this kind of network is, but there's something there, and I think this would help in trying to uncover what that is exactly. Because what we can do with the images is we can like scale up the analysis, and we can just kind of generate topologies, and we maybe even they're not even labeled, but that's not really what's important. What's important is that we have the centroids, we have their relative locations, and we have some criterion to sort them on, and we can generate topologies that way. And so we can actually look at some of these questions about what kind of a network it is. And so it has a lot to do with cell division and where the cells go. Again, like with the EPIC data set that we had, the limitation was that you have the cells are kind of averaged in their kind of final location in development, and then they divide again and they move. So they're moving around, but they stop at a certain point for a while, and then they divide again. So they might migrate a little bit and then stay somewhere for a while and then divide again and form eventually some anatomical structure in adulthood. With C. elegans, you have the labels, but the labels are only part of the story. The real story is where the cells are going. So when we talk about like, how the cell sort of changes it, or how the embryo changes its shape, part of during development. Part of that is these cells moving around, differentiating and positioning themselves with respect maybe to other cells, or in arraying themselves into structures. So we don't really know exactly how that happens, but we can look at like the patterns that they form, and that might tell us something. So that's, I have those slides. If you're interested in looking at those slides, I can put a link in the chat. You can look it over, or if you have ideas about that, you can let me know. Also, we have presentations which are on the website, and we can go over that. So that's that, and then the Bacillaria behavior papers. I want to go over those quickly. So this is switching gears a little bit to Bacillaria. We haven't talked about that in a while. We have the Digital Bacillaria project, which was something that started in 2019. This was the thing that resulted in the Ujjwal was involved in that, and so was Dick. Deep learning analysis of Bacillaria. So you have these diatoms, which are marine microorganisms, and in this specific type of diatom, they live in colonies, and the colonies are sort of have this coordinated movement. What we did in the first paper was to analyze these phenotypes, try to figure out the shape and sort of the shape parameters, and build like these digital models of these colonies. So you have all these cells and their shape. And then we also did some biomechanics, which we don't have images of here. I didn't really up this with respect to that work, but that paper is available. This was Thomas Harbeck, who is a diatomist in Germany. He's been doing a lot of stuff with diatoms and looking at their movement. So he's been doing this thing. He did the biomechanical analysis of this. So that's what the digital Bacillaria is, to remind us what it is. The next step from that, though, is to come at this from a behavioral standpoint. And so this is what these two papers are. And Jesse expressed some interest about this in the last week. So there are two papers, working papers right now. One is this is the one that was submitted as a proposal, the psychophysical world of motile diatom Bacillaria paradoxa. This should be italicized, but that's just the formatting here. So psychophysics is an area of like psychology where there's this measurement of different things in the environment, like physical things in the environment. The nervous system sort of takes in information and operates on in a lawful manner. Beyond that, I don't want to get too much into what psychophysics is right now, but the idea is that there's this psychophysics of behavior, and this is something that you might call this is just something you see in humans. Well, it turns out that you see it in a lot of different nervous systems, but you also see it perhaps in bacteria. I had a citation of a paper where they looked at this in bacteria and in slime molds, things like habituation. So this is psychophysics. This is something that is probably most germane to like sort of unifying the behavior of something like Bacillaria. It doesn't have a nervous system with these sorts of ideas. And so this paper is about going through and looking at maybe what a Bacillaria colony does in terms of its movement that mimics cognition or mimics like a nervous system. So Bacillaria are it's this colony of microorganisms. The different cells, they move relative to one another. They're actin filaments that facilitate this movement. So it's a lot like what muscle does in animals, in animals like, fishes and humans and all sorts of more complex organisms, you could say. But they may have this set of rules. They may actually act like a nervous system in some ways. And so there are these different models of behavioral regulation we can test. we can think of it maybe as more of a Hebbian mechanism, which is something that, if you've heard of the dictum, wire together, fire together, that's what that refers to. So it doesn't require neurons, but it's like it requires coupled systems. And then there's pseudo-intelligence. So maybe this is sort of like, a null hypothesis. Maybe there's just something that looks like behavior that isn't really behavior. And so we've considered that as well. And so there's this literature that we're drawing from as well on aneural cognition. So this paper by Michael Levin and Beluska on having no head, cognition throughout biological systems. They talk in this paper, and there are a couple others, about this idea of non-neuronal cognition, that cells actually produce cognitive behaviors that are, not generated by a brain, but they look like they're generated by a brain. And so this table kind of goes through that a little bit. This table needs to be worked out a bit more. But you have this instance of behavior, which is, if you look at something, does it look neuronal or non-neuronal? And then it's generated by some process underlying that behavior. So, but then that can also be neuronal or non-neuronal. And so Bacillaria chains fit somewhere in here. So, for example, a non-neuronal looking instance of behavior that's generated by a non-neuronal process is the steam engine. It's just an example. Something that's generated by neuronal processes, but that also looks neuronal, could range from like the brain to something like an ant colony. And so, there are differences in how those look, but qualitatively, that's their category. Cell migration is non-neuronal, but it's, maybe I have this wrong. Cell migration has a certain, there's the cell doesn't have a brain, but the instance of behavior is, maybe I have this in the wrong category, but this, like I says, table needs work. But the basic idea is that you have instances of behavior, and they're generated by some process, and you can match those up. And so this paper, this is just a proposal, so there's a lot of work to be done in this. It's basically about interpreting the behavior of the Bacillaria to see if it, what it fits into it. Is it, maybe it looks intelligent, but is it really intelligent? And we can consider those things. So that's one paper. The other one that I have that kind of been working back and forth on is this collective pattern generators. So this is something that isn't really tied to any obligation, but this is something that, so there are these central pattern generators that exist in, say, even like C. elegans nervous system. And so the C. elegans nervous system has these, neurons that fire in a tight circuit that's called the central pattern generator. They fire rhythmically, and they produce these patterns, like maybe like tail movement or body oscillations, something that's rhythmic. And so that's something that we see in Bacillaria as well, except that it isn't generated by the central pattern generator. So the idea that is presented here is this idea of collective pattern generators. And so this is something that you get from sort of, electrical activity, or even like something like actin filaments doing their thing, but they're sort of integrated into a system where there's a collective behavior that's generated. And so we know that collective behaviors can look quite intelligent. They're coordinated, but they're coordinated at a higher level, so we don't really understand too much about that. We can talk about aneural architectures, so we just talk about how Bacillaria doesn't have a brain, but it's generating these behaviors. And then how to model this. So this can be modeled using a series of sine waves, other types of functions to represent the movement of the cells. And this goes back to the data that was collected earlier on Bacillaria. And also how to map these measurements to the data set. So we can do a simulation of the movement, and we can look back to the data, and we can from that we can get some information about how this sort of collective pattern generation works. We can also look to other types of central pattern generation. So there's a data set, CPGs in a stick insect, which actually is a stick insect is an insect with about 6 legs, and it walks along, and when it moves its legs, it generates these the movement is generated by central pattern generators, but they're coordinated because they have 6 legs. And so I don't really have much to share on that data set, but that's there, and they consider in that paper how those CPGs are coordinated. They don't use the term collective pattern generator, but I think this would lead into this idea nicely. So you have these simulations of sine waves, and this Bacillaria colonies are basically a bunch of overlapping sine waves in time. So they exist with relation to one another in different phases, and you can also model them with noise, and you can evaluate the behavior, and then that would be that. I don't really know, this isn't really tied to any sort of book chapter or anything, but that's another thing. This might end up becoming like one paper, or the COPGs might be included in the first paper. Not really sure how that'll work, but I just wanted to give people, is it a gate in a mammalian brain? You mean central pattern generation? Or I don't know. I think gate does have like a central pattern generator. So there's a whole literature on this. I know for sure C. elegans has them, the bona fide, and a lot of insects have a central pattern generator. I think people have hypothesized that that animal movement, animal walking, is generated by a CPG. So I mean, and then in like humans, it's modified by all sorts of other things. So Jesse said, "Thanks for the overview." You're welcome. So I go back to the. So Dick came to me a couple weeks ago and asked me about building a bibliography of all of the work that DevoWorm has done, and so I said yes, that's a good idea. So I sent him a bunch of papers. I sent him a list of the papers with presentations and posters, and then also some of the papers that have been presented in this group. Just anything that we've kind of hit on that looks of interest to the group. So this is something that is ongoing. I'll be curious to see the bib. Yeah, so that's good. If you're interested, you can get in touch with Dick or myself. Jesse seems interested in it. It's just something that I think is good as an organizational tool, and we can link it into the DevoLearn stuff as well, because this is, the DevoLearn stuff on the one hand is like a lot of image processing and everything, but we also, as we pointed out today, we have a lot of things that we've been doing that kind of fit into some of the exploration of the data. So, it can generate a model like a network, but, maybe people have already done this type of work in the group, or, outside the group even, we might want to have some sort of collective knowledge about that. And it's a little less than a Google search. It's a little more concentrated. Yeah, so it may start this week. Okay. Yeah, so let's keep revisiting that. That's good. Susan said the cerebellum creates gait or walking motion. Yes, cerebellum has a architecture of we've talked about this in our other group, where it's like a internal model of the environment and it generates motion. So the cerebellum isn't a central pattern generator, but it does mediate human movement. I was thinking, more along the lines too of like fish is swimming, things that like your heart rate, things that create a rhythm, that require sort of a rhythmic pattern to be there, So it's an interesting topic. Yeah, this is the EndNote basic. So Dick is going to create this in EndNote. So EndNote is bibliography software. People are, I know a lot of the students here are like always doing papers to read. And I know Susan probably knows this, and Jesse might know about this. Dick definitely does. But, you have to have some way of like organizing it and, outputting the references in a format that you can like, manipulate. Because if you get to the point where you're dealing with like hundreds of references, this is where bibliography comes in handy. And so Dick has, I think, a bibliography of hundreds of thousands of papers. He has a master bibliography, and then he sliced it down into different categories, which you can do with a bibliography software. So he's going to build this. Yeah, the commercial version is EndNote 9. He's going to build this in EndNote. And EndNotes, I think you can also use the EndNote files like in things like Mendeley. I'm not really sure the cross. The cross. But that it's basic. It's very good. You'll be able to. It'll be compatible with EndNote, and then we can do things with the references. So that'll be a good thing to have. So I know this is the top of the hour. If you can stay, if you can't stay, that's fine. But if you can stay for a bit, I'd like to go a little bit into the. I don't have it open anymore. I was going to talk a little bit about the. I was going to talk a little bit about the chaos measurements. So Jesse says, is DevoWorm the correct use of capitalization, or is that not yet set? I think that's probably a good stylistic presentation with the capital L, just because we have the DevoWorm with the capital W. But I don't know if it's. I think if you don't use the stylized L, people understand what it is. I mean, it's. But I think we'll probably stick with this stylization because it's. I don't know. It's like two different things, and you're bringing them together. What do you think, Ujjwal?
So I think like what our library name is all in small caps. So if you are using it in the Python version on the terminal, so it is all in small cap. But yes, like for the logo and the name of the repo, I think like they will learn is something like I don't know about like US much, but in India like we used to like make the alphabets capital on which we want to focus on. So that's the reason like why I and Mayukh as well have made like L of capital. So, like I guess it can be like discussed if it has is any sort of problem.
Yeah. Okay. So let's see. Let me stop presenting for a minute. I'm going to pull up those papers again from the. I'll pull them up from the history here. That's not the right one, anyways. So now I can pull back up the screen share here. So Susan, I think last week sent me a couple of papers on chaos, and this is the. Let's go back to that. So here it is. Okay. There we go. And so these papers are good. We have this thing ongoing with the measurement project, or the idea of biological measurement. I know that a couple of us have expressed interest in this, and it's a sort of a sub interest area where sort of like biological measurement and formulating things. I can't remember what the name of the thing is on the task board, but that's not really important right now. If we get into the papers, she sent 3 papers on this. If I can get into them. And they basically lay out, like Susan was saying earlier, it basically lays out this model of like measurements of dynamical systems. It also lays out this way to distinguish between sort of deterministic behavior and chaotic behavior. So like if something is deterministic versus chaos, there's this whole literature about the edge of chaos, which has to do with like systems that exhibit sort of fluctuating behavior that is sometimes it's almost random, but it's not quite random. I think if you George Mikulovsky did a presentation on something called complexification, which is this idea of like how a system can go from like complete determinism to complete randomness, but there's this sort of phase in the middle where you have this sort of potential for fluctuation, and maybe like it enables organization and things like that to emerge. So white noise, brown noise, pink noise. Yeah, so there are different types of things that there's this role of noise in systems for one thing, and systems can be noisy in different ways. And so we think of white noise, which is like the static that you see on maybe like a television screen or a computer screen. And that kind of noise we think of as just kind of synonymous with randomness. But there are other types of noise like brown noise and pink noise that have like this, what they call 1 over F structure, which is, it's an exponential distribution, which means it's, not a normal distribution. It has events that occur in time that are larger than other events. So, it can, right, brown noise is like Brownian motion. So it can do things like, so it's not just randomness, there's some structure to it. So if you think of Brownian motion, that's brown noise. Pink noise is like music. And then I think, I don't know what, if you look online and you look up like colored noise, you can get a good treatment of the different types of noise that there are, because I'm having trouble opening this. I don't know why. Let's see. Go back. I'm just trying to open up these papers. It's exceedingly hard to get into the folder for some reason. But, well, maybe we won't present the papers, but we'll talk about it a little bit. So that's the idea, that there's these chaotic measurements that you can, you usually have like a dynamical system or a time series, and you look at that time series and you analyze it in different ways. I know that like Jesse's probably familiar with this from Neuromatch, but they looked at dynamical systems analysis there. And they're, I don't know if they're using chaos. It's a bit old fashioned maybe for the brain, but there's a guy, Walter Friedman, who published a lot of this like in the 70s and 80s on chaos in the brain and things like that. So they did different measurements at the time series, synchronization measurements, and other types of measurements, and they claimed they could observe things in the brain like synchronization and chaos, and at the edge of chaos behavior and things like that. I'm still trying to get into these papers. I can't seem to do it. I'm not sure why. Let me see. Not letting me get in. Oh well. Well, it was a good idea. But anyways, I just wanted to make, Dr. Whithold Kinsner from the University of Manitoba is an expert. So that's your local expert on this stuff. So, yeah, look up this name here. There are other ways we can, learn about it. There are so people, about in the 70s and 80s and maybe into the 90s, people were really interested in chaos theory. And there was a lot of stuff having to do with like visualizations of complex systems where they visualize these attractors or diagrams of trajectories that were loops and things like that. And there's some actually quite impressive space-based diagrams that they called them, and they were very beautiful. But then actually mapping them to a complex system was another matter. And so they do actually use chaos as a measurement for things. Susan pointed this out in the chat. It's a good measurement tool. And it's definitely something that has been, I think it's like one of those things where there was like a peak of interest when it was like beautiful pictures. And then when it became more, it became a tool, people were like, well, it's not that interesting. I mean, there are people who continued on with the research, but continued on. I'm not getting these papers. I don't know why this won't let me in, but Google Drive is really not my friend today for some reason. Well, anyways, I can't get into the papers right now, but I think if you're interested in this area, we should talk about it more in depth in the following weeks. Yeah, strange attractors and the butterfly effect in weather. So, the diagrams that I was talking about are these phase diagrams. If you know anything about strange attractors, you can look up strange attractors on Google and you'll see all these pictures of attractors, 2 attractor points, 3 attractor points, these fancy orbits. Basically, the idea is that the system has stable states in this phase space, and these orbits are the system moving around those points. And so, you might have a stable point where, the system is sort of stable, but the system also exhibits a lot of variation around that point. So, and then the butterfly effect, if you've ever heard of that, is where you have introduce some small perturbation at one point in time, and it has a multiplicative effect to things later in time. And that's a very popular conception of this sort of thing, the butterfly effect, where if you, stamp out a butterfly here in my location, I cause some sort of weather anomaly in another continent because there are a chain of, causal things that happen that lead to this event happening or not happening. And so, systems have a sensitivity to initial conditions. So if they're, and if you change the initial condition, you can change the system's behavior quite appreciably. This limited diffusion aggregation is another interesting concept, which I don't know if we can do without any sort of visual aids. But it's basically this idea, and I think we've talked about it in previous meetings, where you have like the space and you start maybe with a single clump of something, and you have a bunch of things migrating across the field, right? It's like a field of like things that, so you have this thing in the middle of the field and you have these things that come from the external part of the field and migrate across, and they do this at random, and eventually some of these hit the object in the middle where they hit each other and they form a cluster. And over time, these clusters will grow because things bump into them. This is just a random process. And you end up with these structures that emerge from basically just a bunch of random motion. So the things are moving randomly across this field. They collide, they come together, they become stationary or they start to move in a different direction, snowflakes are a good example. They form these patterns that you wouldn't get otherwise. And I don't know. I think we've talked about on the group. I think I've had some. I think I had a couple. I have like a bag of simulations that I have that I use for instruction, and I didn't bring it with me. But I think diffusion limited aggregation is one of those where you have this, and it's better to look at it when you have this. When it's a visualization than when I'm describing it, because when I'm describing is not like that impressive. But if you see it in action, it looks a lot more impressive. So maybe we can put together, I don't know, I was thinking maybe we could put together some simulations. And we have some of those in the education, the DWOW education repository on GitHub. We have some simulations already based on sort of like, sort of more germane to classic embryogenesis, like about a pattern formation simulations. But we don't really have anything on diffusion limited aggregation, and we don't have anything on like chaos. We have a little bit on like attractors, but that part of the repository was just pieced together. So it's it's one of those things where I think we could maybe use our GitHub repositories to build up some more information about that, maybe apply it more directly to like some developmental process. And we could use it either for education or we could use it for this measurement thing that we were talking about. Because the measurement area is pretty open. I'm not quite sure. I don't know if I have it. I'll see, now it's letting me into these papers. Okay. Well, anyways, it's too late for that. I couldn't get into the papers. The Google Drive wasn't letting me in, so that's fine. So let's see. We have one more thing in the chat. Yeah, snowflakes. That was the one we talked about. Okay, so I think that's good. I think we can maybe talk about that in future meetings about the chaos papers. I wanted to kind of go through them, but I didn't really have time, and I couldn't get into the folder. But I think we can follow up on that maybe with something in this measurement area or maybe in the education area. I think the measurement area is interesting because, I think the goal we want to do there is to try to build up maybe some measurement techniques in that area. Maybe like have, some code perhaps to do some analyses, or, even if it's collected from somewhere else, we can kind of host it locally and say, okay, this is a possible analysis for this type of data. We could have simulations, so we could have a lot of things there potentially. And then Jesse and I talked about that once. But yeah, there are things we can do. Yeah, so I can try to find more information on this for you. Yeah, maybe we can find some more information on specific measurements if there's something we want to follow up on those papers, or it doesn't even have to be related to those papers. It could be like something else entirely. Or just, anything that looks like it's developmentally relevant, we could use it. Okay, well, thank you everyone for attending. Is there anything else anyone wanted to say before we go, or? Okay. Well, thank you for attending, and next week we'll have another meeting. Okay, good. Next week we'll have another meeting. If you have anything you want to contribute, let me know. We'll have updates as we did this week. Stay safe, everyone. Okay, you have a picture of 2 DOAs. Well, send them to me and I can bring them up next week. Hopefully Google Drive will cooperate. Okay, everyone.